The DNA model. DNA is a genetic material which is inherited and determines the proteins which will be produced. It is held together by hydrogen bonds between nucleotide base pairs, thymine, adenine, cytosine, guanine. It is also held by a sugar phosphate backbone. This forms the double helix.
Did you know about 90% of our DNA does not have a selected function, meaning it hasn't been selected to play a role in human physiology? The first step of DNA replication is helicase, unzipping the DNA by breaking apart the hydrogen bonds between nucleotides. Helicase unzips the DNA, and single-strand binding proteins hold open what is now called the replication fork. Step 2. The primase stage. Primase is an enzyme that adds an RNA primer to begin replication. With the replication fork open, primase is put into place and begins building from 5' to 3'. Primase lays down RNA chunks in groups of 3 nucleotide pairs. This bottom strand is called the leading strand.
On the top, we see the lagging strand. This must be made in a direction towards the start of the molecule. The DNA up top is made in chunks called Okazaki fragments. Once in place, ligase then bonds the Okazaki fragments together.
With the RNA primer in place, DNA polymerase starts to form new DNA in the 5' to 3' direction on the leading strand. Ba-doo-da-ba-doo, ba-ba-da-ba. Ba-ba-da-ba-doo-boo.
At this point, we see 2 new strands fully completed.
Because these 2 new strands have one half of an old strand, it is called semi-conservative. This has been our video on DNA replication. Thanks for watching.